This video will provide a big picture overview of the physiology of muscle contraction and some neural events that lead to muscle activation. Here we have an alpha motor neuron, which is also commonly called a lower motor neuron or spinal motor neuron, that is innervating muscle fibers. Collectively, the alpha motor neuron and all the muscle fibers that it innervates is called a motor unit. To activate motor units, excitatory input to the dendrites and cell body of the motor neuron is needed. Our motor cortex, via the corticospinal pathway that comprises upper motor neurons, can provide this source of excitatory input and can voluntarily activate lower motor neurons that innervate muscles, eventually causing muscle fibers to be depolarized. Keep in mind that lower motor neurons can receive hundreds, if not thousands, of synaptic connections, whereby sensory inputs from muscles, joints, and the skin act to promote motor neuron firing or act to hyperpolarize or inhibit their firing. If the excitatory input is large enough to cause a motor neuron to reach its threshold for discharge, this will cause action potentials to be generated along the axon, towards the axon terminals of the motor neuron. Let's zoom in a bit more detail and look at the connection between the axon terminal of a motor neuron and a muscle fiber. The connection or synapse between the axons of lower motor neurons and muscle fibers is called the neuromuscular junction. First, action potentials trigger the opening of voltage-gated calcium channels at the axon terminals of the lower motor neuron. This causes vesicles that contain the neurotransmitter acetylcholine to fuse with the membrane of the lower motor neuron axon terminal. Acetylcholine is then released into the synaptic cleft via exocytosis, which diffuses across the cleft onto nicotinic receptors on the muscle. Nicotinic receptors are ligand-gated ion channels and cause a rapid depolarization of the muscle fiber, or what we call an end plate potential. This end plate potential moves away from the motor end plate before activating more superficial voltage-gated sodium channels. The adjacent sarcomere or muscle membrane is then depolarized in both directions away from the neuromuscular junction, and this depolarization travels along the length of the muscle fibers. In other words, action potentials travel along the muscle membrane. But this depolarization of the muscle membrane also travels down T or transverse tubules. T tubules carry muscle fiber action potentials away from the surface and deep into the center of the muscle cell. This causes calcium ions to be released from the sarcoplasmic reticulum, which is needed for muscle contraction to occur. The sarcoplasmic reticulum is a membrane-bound structure found within skeletal muscle and stores calcium ions. But how does calcium ion release cause muscle contraction? Let's first consider the microstructure of muscle. Within the whole muscle, we have many muscle fibers, and within muscle fibers, we have many myofibrils. Within myofibrils, we have many sarcomeres arranged in series. Sarcomeres are the basic functional unit for muscle contraction. Here we are showing one sarcomere where we can visualize the individual proteins or filaments that make up a sarcomere. The thick filaments are what we call myosin, and the thin filaments are comprised of actin, troponin, and tropomyosin. Troponin and tropomyosin are not shown here. There are also bands, discs, lines, or zones that can be labeled on a sarcomere, but I won't discuss them in this video. When calcium ions are released from the sarcoplasmic reticulum, they bind to troponin, which makes tropomyosin move away from active sites on actin. Actin then binds with myosin to form cross bridges, which requires ATP. The myosin filaments, which contain many myosin heads that bind to actin, pulls actin towards the M line or towards the center of the sarcomere. This shortens the sarcomere. When many sarcomeres in series shorten, this causes the entire muscle to shorten and causes movement at a macro scale level. Overall, the process of muscle activation and contraction is known as excitation-contraction coupling. The end plate potential and depolarization of the muscle membrane is excitation, and the actin and myosin interactions, or cross-bridge interactions, are the contraction. So, to voluntarily activate and contract our muscles, this occurs very quickly, in the order of milliseconds. So it's important to keep in mind that the entire process of muscle activation and contraction, from the cortex to the muscle, described in this video, is very quick and allows us to interact with and move within our environment rapidly and with minimal delay. Thank you very much for watching, and please comment if you have any questions.